talk today about a study we've done on offshore seascape factors and its influence on shallow water fish assemblages on the Swedish west coast. So, because I know that not everyone are marine focused here, I'll give you just a brief background on seagrasses, which are our focal habitat. So seagrasses are a marine plant flowering angiosperm, which are important nursery grounds for many species, including economically important fish species such as cod. They provide habitat as well as food sources. They also supply nutrients to surrounding coastal habitats, and they alter nearshore hydrodynamics as well as preventing coastal erosion by trapping sediment in the leaves. So this in a seascape connectivity context. So it's important to understand the influence of seascape configuration on fish assemblages, because this is a critical step in management approaches. And as was mentioned this morning, the oceans are and will continue to be influenced by climate change. And so we're taking our results and trying to think of them in a climate change perspective. So we know that this is causing increased severe weather events, deep water upwellings, as well as species distribution shifts in some cases. So ocean ecosystems are integrally linked through currents causing water flow. This was also discussed. We can see this in larval dispersal, zooplankton, phytoplankton, ontogenic migrations of species, and all of this increases ecosystem resilience through genetic diversity. It also creates connections between marine habitats, and what we're specifically interested in are our shallow water habitats and how they're linked to the open ocean. And we look at fish as our study organism because they are more mobile organisms and therefore can link our habitats. So our study location, as I said, is the Swedish west coast. It's the Bohuslän area, which covers approximately 4,400 kilometers along the coast. It's a special system because it's an archipelago system with approximately 3,000 islands, but also as many as 5,000 small uninhabited islets with very low tidal fluctuation of approximately 30 centimeters a day, but can be more depending on pressure systems. So a bit more about our methods. We had 30 seascapes ranging the Swedish west coast, and I forgot to mention on the previous slide that our study areas cover approximately 100 kilometers. So each seascape was chosen for the central seagrass meadow, and all of them were assessed for the fish community using stereo video cameras. So if you look at the central picture, this is the setup. It's 2 GoPros mounted on a board. These have been calibrated, and they give you a 3D picture. So where the camera lenses overlap, you're able to measure your fish and identify them, and that can be used for life stage assessment. So the data was collected over 2 seasons, both in the summer and in the autumn. And we took that information on fish assemblage and related it to that seascape proximity to factors, abiotic factors on the offshore areas. We looked at specifically latitude, the depth contour from the nautical charts, deep water areas, which we defined as greater than 20 meters because that's the halocline, thermocline area in our study region. Also, the baseline taken from the nautical charts is a proxy for distance to open ocean. The coastline, because as I mentioned, it's an archipelago system, and effective fetch, which gives you an idea of wave exposure, which is important for shallow water habitats. So a little bit more specifics about our methods. Here's an image of the nautical chart. It's not the best image, but it'll give you an idea. I don't know if you can see these. These are pink areas. Those are our specific study seascapes. And this is just to show you that Here we have the baseline that I mentioned, proxy for our open ocean. So that varies depending on which seascape you're in. How far to open ocean is it? Additionally, we have the depth contour, 20-meter depth contour here. And again, you'll see that if you're here, it's a much farther distance to the 20-meter contour line. So what does that do for the fish assemblages? And because it's nice to see, here's an image from the 3D camera systems, just to show you. We have a school of trout coming in from the right-hand side, and in the foreground we have some small gobiidae species as well. So all of these are captured in both camera views and therefore can be measured and identified. A bit more specifics about the results. So the total abundance didn't change between seasons, but we were able to identify over 11,400 fish from 11 different families and 22 species. And the most abundant species within the most abundant families were actually the same for both seasons, in the summer and in the fall. And the two-spotted goby was what we had the highest abundance of. Woo! And its numbers went up dramatically from 1,400 to almost 5,000 in the fall. So a bit more of the results. We took the 5 most abundant families and did analysis on those. So we have, from left to right, we have members of the cod family. We have sticklebacks, if anyone's familiar with those, goby species in the center, wrasse fish, and then pipefish. And again, you can see this is fish density per square meter taken from the cameras. And the density in the fall increases to almost 300 individuals per square meter of the gobies. And some of the other fish are increasing as well, though some decrease. So of all these measurable fish that we had in our cameras, the total abundance was comprised of 56% juveniles during the summer compared to 72% juveniles in the autumn, though these differences weren't significant between the seasons. And for both the summer and the autumn seasons, the greatest number of measurable individuals came from this codfish family. So that's a little bit about the fish community, but now how is the fish community related to these offshore factors? So I won't go through all of these, and unfortunately the font isn't so great, but here we have distance to our northernmost latitude. And all 5 fish families are influenced by distance to the northernmost latitude, which means that as you're higher in latitude, farther north in latitude, you have a higher abundance of fish species. Additionally, we have 4 of our 5 fish families here that are influenced by distance to deep water, meaning that the closer you are to deep water, the higher abundance you have in fish. So again, increasing abundance of all families as you increase in northern latitudes. And what other studies have shown, put this in perspective, is that there are range shifts of species. We didn't study that directly, but this is related, and it's been documented in both the Northeast Pacific as well as in the North Sea, but many other regions as well. And 4 of our 5 families, including more stationary cryptic species, are positively correlated with deep water. They like the access to the deep water areas, which is likely related to the increase in primary productivity that you have there. So this has been shown in many areas. Deep water brings in well-oxygenated, nutrient-rich water, which is quite good for fish and fisheries. So there's been a study showing that cod tend to avoid areas within this vertical water column that we've talked about with very high currents. And what we see is that our cod family is negatively associated with distance to the open ocean and distance from the archipelago, but positively associated to this deep water. So they like the access to the well-oxygenated deep water, but they also need well-protected habitats, which we have within our fjord system on the west coast. So there is a strong coupling between the benthic and the pelagic zones. And understanding this inshore-offshore connection is very important for management. We also know that species have specific reactions or needs when it comes to dispersal ability, life history, and tolerance to environmental conditions. So we can expect that as environmental conditions change with a changing climate, these species will also react differently. So it's quite important to understand these differences before we see such a large change in order to better target our management approaches. So thank you for listening. Are there any questions?
Thanks, Diana. Really interesting marine ecology methods to understand what structures are fish assemblages. Have you got any questions? Jeff?
A really interesting talk, like really very interesting. I was just wondering, and I know nothing about fish at all, so if I'm